Why are genetic mutations important in bladder adenocarcinoma? Imagine a tiny change in the genetic code of a cell that can set off a chain reaction leading to cancer. That's what genetic mutations are, alterations in the DNA that can influence how cells grow, divide, and die. In bladder adenocarcinoma, these mutations are especially important because they help us understand how the cancer starts and how it behaves. Bladder adenocarcinoma is a rare type of bladder cancer that develops from glandular cells in the bladder lining. Most bladder cancers are of a different type, called urothelial carcinoma, but adenocarcinoma shares many molecular features with other bladder cancers. These features include genetic mutations that are not inherited but acquired during a person's lifetime. These acquired mutations often happen because of environmental factors like smoking or exposure to certain chemicals in the urine. Certain genes are frequently mutated in bladder cancers, including adenocarcinoma. For example, the gene called KDM6A produces an enzyme that helps control gene activity by modifying histones, which are proteins around which DNA is wrapped. When KDM6A is mutated, it can disrupt normal gene regulation, allowing abnormal cells to grow. Another gene, ERBB, is part of a complex that helps open or close DNA to control gene expression. Mutations in ERBB can impair the cell's ability to repair DNA and regulate genes properly, leading to cancer development. Genes like the MLL family, which also modify histones, are involved in activating genes necessary for cell growth. Mutations in these genes can cause cells to grow uncontrollably, contributing to tumor formation and recurrence. Tumor suppressor genes such as TP53 and ERBB act as brakes on cell growth. When these genes are mutated, they lose their ability to prevent abnormal cell division, allowing cancer cells to survive and multiply unchecked. On the other hand, genes involved in growth signaling pathways, such as PIK3CA and the ERBB family, which includes EGFR and ERBB2, can become overactive through mutations or overexpression. This overactivity promotes aggressive tumor behavior by encouraging cells to grow and avoid death. Understanding these genetic mutations is vital because they help explain how bladder adenocarcinoma develops and why it can be so aggressive. They also serve as important tools for doctors. For example, testing for specific mutations can help predict how the cancer might respond to certain treatments. Some targeted therapies are designed to block the activity of mutated proteins or pathways, offering more personalized and effective treatment options. Additionally, knowing the genetic makeup of a tumor can help identify which patients might have a better or worse prognosis. It also reveals the complexity within the tumor itself, known as tumor heterogeneity, which can influence how well treatments work and whether the cancer might come back. In summary, genetic mutations are the driving force behind bladder adenocarcinoma. They disrupt normal cellular controls, promote tumor growth, and influence how the cancer behaves and responds to treatment. Recognizing these mutations not only deepens our understanding of the disease but also opens the door to more precise, personalized approaches to bladder health and cancer care. Always remember, if you have concerns about bladder health or cancer, consulting a healthcare professional is essential for personalized advice and treatment options.